Welcome to the first RNA Collaborative Seminar of 2021. Today's seminar is hosted by the National Cancer Institute RNA Biology Initiative. I'm Sandra Wolin, and I head the RNA Biology Initiative. Today's speakers come from one of our sister institutes here at the National Institutes of Health, the National Institute of Diabetes, Digestive, and Kidney Diseases. Our first speaker is Nicholas Guydosh. He's a Stadman tenure-track investigator in the National Institute of Diabetes, Digestive, and Kidney Diseases. He earned his bachelor's degree at Harvard University and then a master's of philosophy at the University of Cambridge. He earned his PhD at Stanford University working with Stephen Block, and then carried out a postdoctoral fellowship with Rachel Green at Johns Hopkins University. He joined the NIH in 2016, and there will be a question and answer after each talk, so please write your questions in the chat. And with that, I'll give you Nick.
Okay, well, thank you very much, Sandy. Really appreciate the opportunity to speak here today. And I'll be talking about a couple different projects using modified versions of ribosome profiling to look at ribosome-centered RNA quality control pathways. And so I'll first be talking about detection of stalled ribosomes using disome profiling and triggering of the RQC pathway. And that is work from a postdoc, Cezanne Maiden, in the lab. And then I'll be talking about nonsense-mediated decay and using 4DS profiling to detect those events in cells. And that's work by a research scientist in the lab, David Young. So we all know translation begins when the 60S and 40S subunits assemble at an AUG codon, and then the ADS ribosome can then elongate through the message. At the stop codon, termination occurs, and the peptide is released, and the two subunits have to be recycled. And there's a number of reasons that the elongation may not be exactly smooth. It's known that damage to the mRNA can slow down the ribosome. It's also known that premature polyadenylation can slow the ribosome because poly-A itself has been shown to slow the ribosome. And it's also thought that the positively charged polylysine in the exit tunnel, which is negatively charged, can contribute to this as well. And so the question that arises is that stalled ribosomes occur and they need to be recognized and resolved. And the question is, how does the cell do that? And one very obvious way is that when a ribosome stalls, an oncoming ribosome upstream can collide with it, forming a ribosome collision. And we call this a diribosome or a disome. And there's structures now have been shown of this by cryo-EM, and they have a unique interface between the two 40S subunits that the cell can recognize. And there's even now a structure for a trisome complex, and so you can get higher order collision complexes forming. So these collisions are detected, and this kicks off a whole number of events. And one of the sensors that detects ribosome collisions is the E3 ubiquitin ligase HEL2, otherwise known as ZNF598 in higher eukaryotes. And Jonathan Weissman's lab sort of called this constellation of events the ribosome-associated quality control pathway, and it leads to degradation of the peptide, mRNA decay, and ribosome recycling. And so certain RQC targets are well characterized. If you create reporter constructs with 12 or 20 lysines in them to sort of mimic these premature polyadenylation events, you can evoke a very strong RQC response. But one of the questions is, when does this normally happen in the cell? Can this happen at some sites where endogenous ribosome stalling occurs? For example, polyproline is known to slow down the ribosome. Rare codons have been shown to slow the ribosome. As well, if you run out of charged tRNAs due to amino acid starvation, that can also stall the ribosome. And traditionally, amino acid starvation is thought to trigger the integrated stress response. And I'll come back to that later, but the question was whether some of these other sort of everyday stalling events are evoking an RQC response. So to address this question, do collisions form during normal translation? My lab has used an approach called disome profiling, and this was actually pioneered by Sandy Wolin, who's our host here today, in Peter Walter's lab. And then a couple decades later, Nick and Golia in the Weissman lab used a high throughput version of ribosome footprinting to look at translation globally. And what we're doing here is actually a modified form of this to look at these ribosome collided complexes. And so what you can see here in yeast is that we carry out an RNase-1 footprinting that chews away all of the exposed mRNA between ribosome complexes, and that gives us trisomes, or actually monosomes, disomes, and trisomes, and I'll come back to the 40S a little bit later on in the talk. And we can separate those out by sucrose gradient sedimentation, and we can take our monosome, disome, and trisome peaks off of this, extract the cDNA and send that out for deep sequencing. And this is typically what the monosome footprints look like. Generally, it's fairly even coverage across the coding sequence, though slowdown events do tend to cause some accumulation. Our expectation, though, is that if collision events are occurring, these should result in very distinctive peaks in the disome or trisome profiling data. So we wanted to ask, where were these collisions actually enriched in everyday yeast cells? So here's a couple example genes where we thought we might see some of these events. This is HHO1 and PWP1. And you can see there's a run of 3 lysines and 6 lysines. And in the monosome data, you don't really see too much happening. It's fairly even coverage. But in the disome data, you can see a strong peak on both of these. And in the trisome as well, indicating that there are ribosome collisions happening on these lysine sites, as you might expect, but you don't see it in the monosome data. The question then was, do ribosomes collide at other sequence motifs? And so we also looked at runs of proline and glycine as well, which are also thought to slow the ribosome. And sure enough, we could see accumulation of disomes there. And so disome profiling is really a sensitive assay for these ribosome stalling events. So then the question was, are these collisions detected by the RQC pathway? Are these sort of everyday events triggering it? And so what we did to test that, and going back again here to HHO1 and PWP1, was to knock out that E3 ubiquitin ligase HEL2. And sure enough, we could see a change in these peaks. They actually went down in both of these cases. And to look at this more globally, we computed the number of reads at each motif and divided by the total number of reads in the gene. And that kind of creates something that we call a pause score. And larger values of a pause score will tend to show where the disomes are accumulating. So we did this for every tri-amino acid motif. And you can see here in the wild type cells that we tend to have, these are the strongest disome events. You can see here in this plot, each dot represents one tri-amino acid motif. And in the HEL2 knockout, on average, these tend to go down. And so disome accumulation, strong disome accumulation decreases when we knock out HEL2. So to sort of put this together, we think that when disomes form, HEL2 ubiquitinates the 40S. And one model that we've entertained is that this creates some sort of intermediate that the cell can then decide whether to continue on to RNA decay and other pathways, or allow that disome to resume translation. And it's known when you knock out HEL2, other labs have shown you tend to get more downstream translation occurring. So we wanted to check just how many of these events lead to RNA decay. So we did RNA-seq on a HEL2 knockout and a wild type, and you can see we had pretty good correlation. There really wasn't that much difference, suggesting there's not a lot of RNA decay happening. But the few outliers we did see turn out to be targets of the GCN4 transcription factor. So as you're probably aware, GCN4 is part of the integrated stress response. It's sort of a global transcription factor. And just to take a step back, when ribosomes stall, and traditionally this is due to uncharged tRNAs being present during amino acid starvation, it's known that the kinase, GCN2, will phosphorylate eIF2 alpha, one of the core initiation factors, leading to a global reduction in translation initiation, though some transcripts like GCN4 can escape that and are actually upregulated. And it's been known, several groups have suggested that GCN2 interacts with the ribosome in some way, but it hasn't been entirely clear how that might happen. So this was really interesting that we're seeing this pathway getting triggered by simply knocking out HEL2. And to confirm that that really was phosphorylation of eIF2 alpha, we checked for that by Western, and we could see sure enough that eIF2 alpha phosphorylation does increase in the absence of HEL2. And I'm not going to show it here, but we've also knocked out GCN2 and show that it is going through the GCN2 kinase. And so this suggests that there's actually multiple sensors for disomes, both HEL2 and GCN2 can detect these. And the question was, what we're seeing is that GCN2 is detecting disomes in the absence of HEL2. Can HEL2 detect sort of traditional targets of the integrated stress response, that is amino acid starvation? So we starved our yeast cells of amino acids with a compound 3-AT. And you can see here a average or metagene plot of disome reads on His codons in the presence of 3-AT. And sure enough, you can see a really strong peak there on the His codon. So disomes are forming under amino acid starvation. And we wanted to ask, was HEL2 actually recognizing these sites? And so we did mass spec of RPS3, which is one of the targets of HEL2. And you can see in the absence of HEL2, we see almost no ubiquination of RPS3. But in the presence of 3AT, we see a 10-fold increase. And so what this is telling us is that HEL2 is recognizing these traditional integrated stress response substrates under amino acid starvation, and it is also forming disomes. So to put this together, what we think is happening is that we have formation of disomes due to any number of conditions, and that these are recognized by HEL2, and there may be some kind of stabilization that happens that allows the cell to kind of integrate other information, such as how strong the stalling event is, and decide whether or not to send these to RNA decay. It looks like under normal conditions, a lot of these are not decayed, and so you may have cases where translation can resume. But at the same time, GCN2 is also detecting these disomes and leading to activation of the integrated stress response. And what's really interesting about this is that we know the integrated stress response globally turns down translation initiation. So this is going to reduce overall traffic on mRNAs, and you might expect that's going to reduce the occurrence of disome formation. So that might actually explain why when we knock out HEL2, we see a reduction in disomes. It's because we're actually turning on the integrated stress response to some extent. And in that way, RQC may also be serving as a buffer to prevent unnecessary or chronic activation of the ISR. And I'll point out, as we were doing these studies, several other labs were finding the same thing. So Colin Wu, who's also here at NIH, did this in Rachel Green's lab. Hani Zaher's lab has also found this, and there's a really interesting structure available from Daniel Wilson's lab and Roland Beckman on bioRxiv that really nicely shows exactly how GCN2 is detecting these disomes. Okay. So now I'm going to shift gears here just a little bit and talk about nonsense-mediated decay. So NMD is another translation-dependent RNA decay pathway. And as many of you know, the way that it works is that if there's a premature stop codon present in a transcript, that transcript will be decayed. And this is reliant on the factors UPF1, 2, and 3. This is an important process to understand, not just because this is obviously important if you make a bad transcript, but to understand what the usual substrates are of NMD. So much as we were looking at what are the endogenous substrates of RQC, we were very curious about this question in NMD. And many people have been asking this question recently, in part because NMD is now an important drug target. So it's important to know what it's targeting under normal conditions. And a recent study that came from Alan Jacobson's lab looked at this and found that over 900 RNAs are degraded by NMD. And a lot of these, most of these, didn't have a really obvious premature stop codon. So it suggested there's a lot of NMD happening, but it wasn't immediately obvious why that would be. And many labs have suggested similar ideas for why this might be, and they all kind of relate to the idea that there are cryptic translation events that are happening, open reading frames or in different frames that could be explaining why there might be a premature termination event happening, leading to degradation of some of these transcripts. So we wanted to apply 40S ribosome profiling, which gives us a very, we've developed it to give a very high resolution view of where cryptic translation is happening, to see if we could try to understand this question as well. And so I'm going to speak to an example gene here that's part of that list of the 900 or so transcripts in yeast that are targeted for NMD. One of them is the transcript for DAL5. DAL5 is a transporter of allantoate, which is a nitrogen catabolate, and it's needed for growth on poor nitrogen. So I'm going to show some RNA-seq data here, and apologies here. This we sort of the cityscape view here is due to us using a TrueSeq kit and getting a little bit of bias in that we're redoing these, but you can see quite clearly the main effect here, which is that under poor nitrogen conditions, the canonical promoter of DAL5 turns on and you get the transcripts. But what's interesting here is that there's also a longer isoform present that you can see both under the rich and poor conditions. And it turns out, and there's the long transcripts, when you do this in a UPF1 knockout background, so NMD is inhibited, you can see that this longer transcript isoform comes up. So it's the long form that's actually under NMD. And even in the absence of UPF1, you can still get some activation of the canonical transcript. So the question for us why is DAL5 long isoform under NMD control? So to do this, we've developed another modified ribosome profiling approach, and this is based on something referred to as TCP-seq, which is a form of 40S subunit ribosome profiling, and this was first reported by Thomas Price's lab. And one of the key differences here, unlike the other ribosome profiling methods, is that this requires 1% formaldehyde cross-linking because the 40S subunits are a bit more fragile and they have to be cross-linked to the mRNA. So then we can select both the 40S and monosome fractions from the sucrose gradient, much as we did before, and extract the cDNA and perform deep sequencing on it. And you can see we get fairly even coverage across the open reading frame as usual we expect that for the monosome footprints. But the expectation for the 40S reads is that we should really only see within the coding sequence only two places. We should see them at the start codons and at the stop codons. And that's because at some point you have a 40S sitting on the start codon awaiting the 60S to join it. And then at the end of translation, once the 60S is recycled, the 40S should be there for some time as well. And these two peaks should really offer a nice way to identify open reading frames. So we wanted to find out, could we find non-canonical translation events using this kind of method? Okay, so here's some 40S ribosome profiling data. This is a metagene or average analysis, and these are wild type cells. And so you can see we do get a peak at the main start codon, as you'd expect. And at the stop codon, it turned out we really didn't get a very strong peak. And this is actually because it turns out 40S ribosome recycling in other work my lab has done is shown as a very efficient process. And as soon as that 40S is left there, it is very quickly recycled. So we have a couple tricks to sort of get around this. One was to enhance the size of the start peak is to knock out RPL11B, which is one of the two copies of RPL11. And this creates a shortage of 60S subunits in the cell and then increases these peaks on start codons because that 40S ribosome is sitting there waiting to initiate much longer. So we can actually augment the peak here at start codons by knocking out RPL11B. And we can do something similarly at stop codons. We can knock out, my lab and others has shown that the 40S ribosome recycling factors in yeast are known as TMA64 and TMA20, and in mammals, this is MCT1, DE&R, and EIF2D. And if you knock these out, you dramatically slow down 40S ribosome recycling, and you can get a very strong peak at the stop codon. And so these serve as fiducial marks that very nicely mark the ORF boundaries. And then if we plot these, either the 40S start peak height or the 40S stop peak height against ADS ribosome profiling coverage, we see that these are correlated. And so this tells us that these are predictors of the amount of translation happening in open reading frames. And in some ways, this is similar to use of Harringtonian or lactobamycin runoff type ribosome profiling. But what this gives you is really both ends, which we think is a really nice advantage for finding translation events that aren't immediately obvious in normal ADS ribosome profiling data. So now going back to the example of DAL5, recall we see this long transcript isoform in RNA-seq in a UPF1 knockout background, and it's subject to NMD. And if you look at ADS ribosome profiling on it, you can see some footprints that are sort of at the 5' end. It suggests these might be upstream open reading frames, but it wasn't entirely clear to us. So we then did the 40S footprinting on this, and you can see very clearly a bunch of peaks corresponding to start codons, and as well as some peaks corresponding to stop codons. And the idea here is that these really do look like UORFs, and therefore these UORFs could be causing NMD on this long transcript because these are termination events that are very far away from the 3' end of the message. And if you zoom in on these, you can see there's actually 4 of them. And we have a very high precision view for where these are. We can see there's a start codon here on UORF1, stop codon on UORF1, start codon on UORF2, and the stop here on UORF2. And we can see the same thing here in UORF3, though it's there's less translation happening because a weaker start peak and a weaker stop peak. And on UORF4, we really only get the stop peak. which makes sense based on the scanning model of initiation. You get your 40S ribosomes are sort of gradually initiating at these early UORFs. And so it's very clear to us that there are UORFs that are being translated on DAL5, and that's explaining, is probably the best candidate to explain why this is a trigger of NMD. So to then sort of put this together, DAL5 here has two promoters. It has a distal promoter and a proximal promoter. And under normal conditions, the distal promoter is primarily being used, and this results in an mRNA with a bunch of UORFs that get translated and a main ORF that is essentially never translated based on the data that we have seen. And so the question is, why would the cell be making this long transcript isoform if it's not going to be at all translated? And a really reasonable explanation for this comes from work from Elgin Nudal and Gloria Brar at Berkeley, who defined the architecture for this kind of transcriptional program as a LUTI. And so what that stands for is long undecoded transcript isoform. And so the idea here is that transcription here at this long or LUTI promoter is actually silencing the proximal promoter through chromatin marks. And so this is actually a powerful way to control gene regulation. And what we've now seen is that a lot of these LUTI transcripts appear to be subject to NMD. And so NMD appears to be needed to further repress these LUTI isoforms. And we find that there's quite a number of these. It was already suggested that LUTIs might exist at least 10% of the transcriptome, and probably there's quite a bit more. And they're involved in a lot of key metabolic pathways, including nitrogen regulation. And what I can show you is, sort of I don't have time to get into it, but we think that probably based on the sort of analysis I showed you, about half of the NMD targets are LUTI-like or have these sort of long isoforms with UORFs on them. And there's a number of other categories for other sorts of transcripts that we see that are subject to NMD. Many of these are the sorts of things that others have shown as well. But we feel the 40S profiling approach has been really a productive way for finding these sorts of cryptic events. Okay, and so I think that's it here. I want to just take a few acknowledgments here before taking questions. I want to acknowledge first the two very talented people who did these studies. So Sezen Maiden, as I mentioned, is a postdoc in the lab who did the RQC work with dizone profiling, and David Young did the 40S profiling. He's a research scientist in the lab to look at NMD. I want to thank the other members of my lab, a really great team who've really given a lot of great feedback as these projects have gone along. I want to acknowledge the proteomics core for helping us do the RPS3 mass spec, the genomics core, and some of the other translation labs here at NIH who've been really great about giving us feedback along the way. I want to acknowledge, again, NIDDK, the intramural program, for support. And if I don't have time to get to your question, please reach out to me by email. I'll be happy to get back to you.
So write your questions in the chat for Nick. Here's a question. Are you able to detect stop codon read-through events more efficiently with 40S profiling compared with ADS?
It's a really good question. So with the 40S profiling, we should be able to see that secondary termination event in our mutant. And so that's a really good point. We haven't explicitly looked for read-through events, but we should be able to see stop codons lit up. We won't see the start codons, of course, because they're read-through, but so we'd lose that. But I think we would still have that that approach. So that's a really good suggestion.
More questions? Type away. So does RPL11 delta, can that be a misrepresentation of an actual upstream ORF? I guess that by deleting the component of the 60S and lowering their level, might that give you some misrepresentations?
Yeah, that's a really good point. I think in these different approaches for trying to find cryptic translation, this is always a concern that you're somehow perturbing translation itself. And so that certainly is a concern. Similarly with Harringtonian-based methods, you get a lot of runoff and you're sort of flooding the cell with ribosomes. And so that I would say there's probably, we don't have the perfect approach yet. One of the arguments that we like this here for this approach is that we're using 2 separate mutations. And so we can use both of those, and hopefully we could cancel out potential artifactual effects like that. We can also just look at the wild type data. We did get a start peak without the RPL11. So we really wanted to eliminate that. We could do that.
Could RQC apply to toxic repeat expansions like Huntington disease?
Yeah, so that's a really interesting question. Is the RQC pathway there to sort of guard against things like this? And if you've got arrested ribosomes, there's no reason, this couldn't be there. I think that's still one of the really important questions right now, is just how, RQC is detecting a lot of things, but what determines just how much it's turned on? And are there ways to circumvent it? And in some of these diseases, maybe there's ways that it gets around it. Otherwise, you'd think it would be sort of helpful in that effect. So that's a really a great question for future thought.
So Marcus Hopfler is asking, what is the relation between LUTI transcripts and cryptic transcripts that also regulate promoter chromatin modifications?
So I think that this is a similar idea. If I understand the question right, I would say the LUTI transcript in some ways is a cryptic transcript. I think maybe I'm taking here the word cryptic maybe suggests that it's not as regulated. I think the idea with the LUTI transcripts is that these are sort of very obvious transcriptional programs where you've got multiple genes in the same pathway. For example, in the nitrogen pathway, we see at least 4 different genes that all seem to be using this. And so I think that it might just be kind of a question of semantics there.
We have a question from Nelson Lau. Are stalled ribosomes always at the same particular amino acid repeats? And are these stalling sequences conserved across kingdoms?
Yeah, that's a good question. So yeah, there are certain motifs that definitely do tend to stall the ribosome. And to some extent, these are conserved, though this can be influenced by other things like for a particular organism, what's considered an optimal codon. And so that may be different. And so in some cases where you are relying on tRNA levels to control the speed of elongation, they may not be. So I would say in some cases, yes, but not necessarily. And I'm actually going to stop sharing. So Katie can get started.
Okay, Nick, are you here?
I'm still here though. Yeah, can you see me?
Okay.
Yeah.
I can see you. Okay, so from Arcana Yara, why does the HEL2 mediated RQC pathway supersede the GCN2 mediated ISR pathway?
That's a really good question. Yeah, what's the order of operations here? Why, are these 2 pathways in competition? Why does it seem, that GCN2 is sort of the backup for that? And that's a really great question. And I think we'd like to learn, and it may, structure may be a really good way to get at that. That's a great question.
And I see from Kelly Dean, do any of the upstream ORFs and LUTI transcripts produce peptides, or are they regulatory events only?
That's a great question. Yeah, I think that gets at maybe a larger question of why do you need NMD on a LUTI transcript? The UORFs do prevent the downstream main orphan from being translated, and I think the UORF peptides is one potential explanation for that. That those UORF peptides maybe are toxic or something, and so actually NMD helps reduce the number of UORF peptides. Alternatively, the idea that those could be functional, I think, is also really interesting as well. So I think that's kind of been a long-standing question: is do UORF peptides have functional roles? And so I think we're thinking about that for sure.
So this is going to be the last question. It's from Astrid Haas. Are LUTI transcripts efficiently exported to the cytoplasm, or do they also lead to decay in the nucleus?
Yeah, I would argue that they're going to be cytoplasmic in order for translation to happen. If they are, there may be mechanisms though to degrade them in the nucleus as well. That's a fair point. If you could figure out how to decay them in the nucleus, that would also be efficient at doing what they need to do in terms of repressing downstream transcription. So that's a great point.
So the remaining questions Nick can answer in the chat after he's done. So we'll move on to our second speaker. That's Katie McJunkin. She is also a Stadman tenure track investigator in the National Institute of Diabetes, Digestive, and Kidney Diseases. She earned her bachelor's degree at Princeton University and her PhD degree from the Watson School at Cold Spring Harbor Laboratory. And she carried out postdoctoral research with Victor Ambrose at the University of Massachusetts Medical School. And she joined the NIH in 2017. So welcome, Katie.
Thanks, Sandy, for the intro, and thanks for the invitation. I'm really excited to share my work in this forum to talk to you guys about our lab's work discovering important microRNA target interactions. So as you all know, microRNAs are small RNAs that are bound by an Argonaute protein, and as part of this complex, they bind to complementary mRNAs and inhibit them at the level of translation and stability. And the seed sequence of the microRNA, or nucleotides 2 through 7, counting from its 5' end, are the most important in target recognition. And so because of this, different microRNAs that are encoded in the genome are together grouped as a family because they act largely redundantly on a common set of target genes through seed complementarity. And also because of the importance of the seed in target binding, target prediction and identification is very difficult. So this is because a perfect match to your microRNA seed will be found thousands of times in the transcriptome, but not all of these sites are likely to be real regulatory interactions. And so one approach we usually take to this is to prioritize those sites that have evidence of evolutionary conservation. But still, this narrows down to a few hundred sites, many of which are probably also not regulated. The conservation in this region could be for other reasons, like binding to RNA binding proteins, which of course would occlude the site from a microRNA recognition. The sites could also be occluded by other contextual factors, such as secondary structure. And on top of all of this, although a seed complement is sufficient for target recognition and repression, it's not necessary. So another common mode of target binding is with imperfect seed base pairing and additional supplemental base pairing in the 3' end of the microRNA. And clash data sets have also shown us that there are additional modes of target engagement as well. So empirical methods like clip and clash are really crucial to supplement in silico predictions, and ribosome profiling and RNA-seq are also gold standard to identify transcripts and transcripts whose stability and translation responds to overexpression or inhibition of a given microRNA. But even if we had a complete list of the biochemical targets of a microRNA, this still leaves important questions from a biological standpoint. So even if we know all the targets of a microRNA, we don't know which of those targets are the most important ones in regulating normal physiology. So another way to frame this question is, in a wild type scenario, all the biochemical targets of a microRNA are regulated and repressed, leading to a wild type phenotype. And in a microRNA mutant, all the targets are de-repressed, leading to some phenotype which could be deleterious. And we don't know which of these targets de-repression is causing this phenotype. So this is the question that we want to ask. And the context in which we're interested in this is in embryogenesis. And there's a number of reasons for focusing on microRNA target interactions during embryogenesis. One is just a basic developmental biology question of what are the holes in our understanding of gene regulatory networks that drive embryogenesis are actually filled by microRNAs that was previously unappreciated. And this stage of development is somewhat understudied because it's not very tractable in many models, like in mammalian organisms. But a second reason for studying embryonically expressed microRNAs is there's some evidence in recent years that in this context, microRNAs may have a different mode of repression, favoring translational repression of their targets over destabilization of targets, which seems to dominate more in differentiated tissues. So only by understanding the microRNA target interactions in the embryo and studying them can we begin to understand how this switch in output of the risk may occur. So the model that we're using, since we want to study early development, is C. elegans, which is a great model for studying early development. It has a completely defined cell lineage and is cheap and fast to grow and transparent for imaging studies. But for the story I'm going to tell you today, the most important feature of this model is the great genetic toolkit, to which we've added CRISPR-Cas9 in the last few years, and that has really opened up the type of questions that we can ask. So again, we want to ask this question: which targets are most important in contributing to a microRNA mutant phenotype? But instead of a cell-based phenotype, we're going to be looking at a whole organism phenotype. And this is a really key difference from previous CRISPR screens, which are pretty much only on cell-based phenotypes, whereas we'll be looking at whole mutant animals to understand contributions to development. And thinking about the last slide, you could probably already predict what our approach is going to be. And that is to go into each microRNA binding site and mutate them one by one and ask, what are the consequences for the phenotype? And so for some targets, we expect that relieving them from microRNA-mediated repression could recapitulate, partially or fully recapitulate the microRNA mutant phenotype, whereas other targets may be de-repressed but not have an overt phenotype. And so in doing this type of screen, we're looking for the essential microRNA target interactions that are required for normal development. And the microRNA family that we're interrogating first is the miR35 family, which has 8 members that share an identical seed. And they do act redundantly, because if you knock out all members of the family, this results in complete embryonic lethality. And having just 1 or 2 microRNAs remaining in the genome completely rescues this lethality, unless it's the least expressed member of the family, in which case we have a hypomorphic phenotype. And this hypomorphic scenario has allowed us to also define some additional functions of this microRNA family in promoting fecundity, so the number of progeny that the animals can make. And when this microRNA family is compromised, so is that fecundity. So in C. elegans, with a strong phenotype like this, we would normally dissect it by a forward genetic screen using chemical mutagen to find suppressors of the phenotype, which would define microRNA targets in this case. But such a screen was done by the Horvitz lab when this phenotype was first characterized, and no suppressors were identified. And so one of the possible explanations for this is that there's more than one target that is de-repressed in a way that causes lethality. And so that makes this microRNA a great candidate for this type of screening, since it's been refractory to analysis by traditional genetic means. And some other features of this microRNA family that make it amenable to this type of screening is that there's only 1 predicted binding site for the microRNA per transcript. And also the seed sequence is GC rich, which just means that the sites that it binds to in target mRNAs contain a lot of NGG, or the recognition sequence required for Cas9 to cleave nearby. So both of these features make guide RNA library design more straightforward than if this were not the case. And I'll mention here that all the work I'm going to talk about today was done by a really talented postdoc in the lab, Bing Yang. So when we set out to do CRISPR at each predicted microRNA binding site of the miR35 family, we realized that we may not always get the type of mutation that we're intending. So if we try to target a wild type 3' UTR where the seed match is highlighted in yellow, sometimes the indel that results from non-homologous end joining after the double strand break will happen to affect the seed match region disrupting microRNA binding and causing target de-repression. And we'll call these seed match disrupting alleles. But in other cases, the stochastic nature of this process may mean that the indel will be outside of the seed match and would not be predicted to affect microRNA binding or repression. And so at an essential site, the seed match disrupting alleles would recapitulate aspects of the mutant phenotype, whereas the seed match intact alleles would not have phenotypic consequences. And at a neutral site, these two classes of mutations will have equivalent outcomes, since not even de-repressing the gene leads to phenotypic consequences. And so we thought perhaps if we do this screen in a multiplexed way and read out the resulting alleles by deep sequencing, we can in fact turn this into an advantage rather than a drawback. So if we impose selection on the CRISPR strains and we select only for wild type strains, then this should select against seed match disrupting alleles at essential sites. So then if we profile all the alleles at a given site, this gives us a metric of the likelihood that a given site has phenotypic consequences. So a low seed match disrupting allele frequency would suggest that there's selection against disrupting the seed match and that this is an essential site, whereas a higher seed match disrupting allele frequency might suggests that disrupting the seed doesn't have phenotypic consequences. So we first had to optimize multiplexed CRISPR screening in C. elegans, which hadn't been done before. And we found that we could multiplex up to 10 different guides and inject them simultaneously into the syncytial germline of C. elegans without losing efficiency of each individual guide. So we pooled all the guides into pools of 10. We had 90 guides, one targeting the binding site in each predicted target of this family predicted by target scan. And there are more pools than you'd expect because we included each guide RNA in 2 distinct pools with completely different sets of pool mates to control for possible synthetic effects of injecting multiple guides together into this syncytium. And the gene editing is going to happen in these germ cells and in the 1-cell zygote. So the resulting progeny will be mutant in the whole body, and these mutations will be passed on to their progeny. So we select superficially wild type strains from each of these injections, 30 wild type strains per guide RNA pool, and then PCR and deep sequence the edited loci from these strains. And again, we're looking for evidence that there is selection against disruption of the seed match versus no selection against the seed match disruption. So the summary statistics from the screen show that most sites had multiple alleles in the wild type population. When the guide RNAs were assayed twice in 2 different pools, the data were strongly correlated. So the synthetic effects of having multiple guides together in the syncytium were really minimal. But we noticed that there were this set of loci where there were not any alleles in the wild type strains that we harvested. And so we wondered, are these actually the ones that we're looking for? And any allele in this site is really strongly selected against. So perhaps we were overthinking it. So we went back to each of these sites and attempted to edit them with a second or a third guide or with different conditions. And in each case, we were able to edit that putative microRNA binding site without phenotypic consequences. So the lack of alleles in the primary screen is actually just due to low guide RNA efficiency and not due to phenotypic consequences. So this again really highlights the importance of having a metric like frequency of seed match disrupting alleles instead of just something like allele number. And so when we plot the rest of the loci according to this metric, we get the whole range of possibilities, and I'll show a couple example sites. So EGL1 is a very high priority candidate because we have 5 distinct alleles in the wild type selected population, none of which affect the seed match region. At this other gene, we had 7 distinct alleles and they all happen to affect the seed match. So we took the top 6 scoring candidates with the lowest frequency of seed match disrupting alleles. And we went back to each of these sites and mutated the 3' UTRs one by one to examine the phenotypic consequences and see whether any of them recapitulated aspects of the microRNA mutant phenotype. And we saw for EGL1 that we saw both aspects of the mutant phenotype recapitulated, both low fecundity and on their own, these 3' UTR mutations cause embryonic lethality. And this is just 2 different alleles at this UTR1 provided by Brent Derry's lab. And I'll note that these other milder effects we did follow up on, which I don't have time to talk about today, but it seems that some of them are not reproducible with additional alleles, and some of them don't seem to be due to loss of microRNA-mediated repression. And so it was really gratifying to get EGL1 out of the screen because EGL1 is a previously known target of the miR35 family. The Duchenne lab showed that EGL1 is deadenylated in a miR35 dependent manner in vitro, and the Conrad lab and Derry labs showed that in vivo, this microRNA represses EGL1 in the embryo or in the germline. And the Conrad lab went further to show what the phenotypic consequences of this are. And so EGL1 is a BH3 only proapoptotic protein. And when it's not repressed by miR35 and Bantam, its expression reaches higher levels prematurely. And this causes programmed cell death to occur too early. And so in this case, this daughter cell is fated to die, but because of the premature buildup of EGL1 in the absence of microRNAs, the mother cell apparently dies instead. And we didn't previously know that this effect was sufficient to induce anything like embryonic lethality because it's very low penetrance and very few cells that are affected in this way. But our work now, which is really enabled because of CRISPR mutating the 3' UTR, shows that this de-repression of EGL1 is sufficient to cause lethality. And because our work is the first to show that, we wanted to be sure that these 3' UTR mutations were causing these phenotypes because of loss of microRNA-mediated repression. And to test that, we made additional mutations at the EGL1 3' UTR. Sorry about that. And showed that if we, instead of deleting the seed match, we make mutations here, we still get the phenotypes, low fecundity and increased induction of embryonic lethality. And if we go back to these strains and CRISPR them again at the miR35 locus to introduce compensatory mutations that restore binding to the EGL1 transcript, then we see almost complete or partial rescue of these 2 aspects of the phenotype. So this is really strong evidence that these mutations in a single 3' UTR binding site are inducing embryonic lethality in this case due to loss of microRNA-mediated repression. And we think this is a really great gold standard experiment for target validation going forward. And what I mean by that is using CRISPR at both the microRNA and the target locus, since both of them are expressed at endogenous levels. So to sum up, we established multiplexed CRISPR screening in C. elegans, and we have some exciting ideas about different directions to take this in the future. We found that mutational profiling at each targeted site was really important in interpreting the results of our screen because there was a high variation in guide RNA efficiency. And we were able to discover biologically important targets, including a single microRNA binding site that, when deleted, induces some degree of embryonic lethality on its own. And this type of screen could be expanded not only to other microRNA families, but also to any set of candidate negative regulatory elements, such as sites for RNA binding proteins or transcriptional repressors. And so you can screen through a set of putative binding sites and mutating each one to see which one recapitulates the known phenotype of loss of function of the protein. And so this is just one example of the many ways that CRISPR is changing the types of questions that we can ask. And if you want to read about more details, this was published a few months ago in Genes in Development. I'm looking forward to expanding this type of screen to other model systems or other microRNAs. I mentioned before that the miR35 family is particularly amenable to this type of analysis. So what sort of challenges will there be that we need to solve for other microRNAs? One is this lack of the NGG in other binding sites for other microRNA families. And hopefully PAMLess Cas9 variants will prove really effective and be able to totally alleviate that concern. But in cases when cleavage does need to be separate from the seed match by any distance, we think it would be important to incorporate this into the design of the analysis, probably binning according to distance between the cleavage site and the seed match. In other models, considering off-target effects of the guide RNAs will be even more important than in C. elegans due to larger genome size and the difficulty or inability to backcross. And our colleague Joanna Vidigal is doing some really great work in this area at NCI, which should hopefully enable for more rational custom guide RNA library design in the future. I mentioned that there's only one predicted binding site for miR35 for target gene, and for other microRNAs, this may not be the case. And so there's a couple different ways to think about it, such as you could just assume that each site will have its own measurable effect on the phenotype, which is consistent with each site being evolutionarily conserved, and also would be consistent with cooperativity that's been observed between many sites. To be more conservative, you could also design bicistronic or multicistronic guide RNA vectors to include in the library to target more than one site at once. And finally, we chose a really strong phenotype to study, but for future screens, it might be even more prudent to choose a very specific phenotype and might increase the chances that de-repression of a single target will be able to recapitulate that phenotype. And so especially fluorescent reporters, which detect deregulation of a particular pathway or gene that happens downstream of a microRNA, might be a really good option. And so I'll stop there and thank the lab. We of course have our obligatory Zoom photo, but I'm nostalgic for the old conference days. And Bing Yang again did all this work. And I'd like to thank other worm labs for strains and discussions, as well as our local community and NIDDK for funding. Take questions.
So thank you, Katie. So we already have questions in the chat. So the first one is from Javier Beaufil-de-Ros, and he says, could the screen have missed targets that have extensive base pairing beyond the seed sequence? Could that be addressed by combining it with AgroClip data? Yeah,
so we are concerned that the screen is still limited by the target list that we choose to screen, of course. And we are thinking about going back and repeating it supplemented with clip data. Another feature of this microRNA is that it's in a different argonaut than is usually studied, since there's more than one microRNA argonaut in C. elegans. So the existing clip data is for the primary argonaut, and we're working on generating clip data for this argonaut to which miR35 family is bound.
We have a question from Chol Kwan. It says, can we get a similar result using guide RNAs targeting the coding sequence in the transcripts?
Yeah, so you can do kind of an opposite screen, I would say. Perhaps in the context of the microRNA mutant, you could inactivate every putative target gene and see if any of the gene inactivations suppress the phenotype or revert the phenotype. I'm not sure if I'm getting that question right, but I think that would be the way to use existing CRISPR libraries that target coding sequences to ask the same kind of question.
We have a question from Nelson Lau. Why can't the EAGLE1 mutants have any mutations in the microRNA seed sequence?
Yeah, that's a great question. So we think it is due to aberrant apoptosis. It could be in these cells that have already been characterized by the Conrad lab to be aberrantly undergoing apoptosis. And the fecundity also could be due to increased apoptosis in the germline. So the dairy lab showed that de-repression of EGL1 causes increased apoptosis in the germline in the presence of radiation, but actually it could be happening even in normal conditions, affecting the fertility.
Here's a question from Michael Oreger. So several groups have recently shown that CRISPR-induced DNA repair outcomes are often guide specific. Some of the guides would be expected to induce only short indels that may not reach the microRNA seed region. Have you found cases where the phenotype of the microRNA target site was not confirmed due to this problem?
Let's see. So we did, yes, there's this data that shows that I think it's the efficiency of the guide RNA. Based on its efficiency, will have different types of outcomes for the repair. We haven't looked closely at that. For the sites that did not validate in the single gene studies, we don't know yet whether perhaps we just weren't stringent enough with our cutoff for validation, since EGL1 was the most strongly scoring gene. That's probably not the case. It's probably true that there's other effects of mutating the 3' UTR that are not microRNA dependent. I'm getting a little off topic, but we haven't looked closely at that, and we do see things that don't validate for at least some reason, but could be a number of reasons.
Okay, any more questions? So if not, we've finished fantastically on time, and I'd like to thank both Katie and Nick for giving gorgeous talks, and see you all in 2 weeks.